So I'm excited to introduce today's speaker, Dr. Nicole Lake. Nicole is the current fourth-year postdoc in Dr. Monko Lake's lab. As you know, it's a very traditional human genetics or genomics lab, and Nicole got her PhD from University of Melbourne, and actually the thesis work on the mitochondrial topic too, and she joined Yale with sort of equivalent NIH F32 postdoc fellowship in Australia. And she continued working on the mitochondrial, and I had some interaction with Nicole early regarding sort of the mitochondrial data analysis. So I was impressed. The research, I think her research would benefit from our clinical audience. She and I just talking. This is a department. It's a hybrid department. I really want to kind of invite or introduce more postdoc or the faculty from clinical or research, basic research to this platform, so we can have a more interaction and benefit for both sides. And in the future, you have someone or even the audience here, you have a topic you want to introduce to this audience, please feel free to kind of email me or email Jennifer, and she's our coordinator anytime. With that, I can let Nicole take the floor, and thank you again, Nicole, for presenting to Grant.
Okay, hopefully everyone can see my screen.
Yeah.
Firstly, thank you, Yonghui, for the invitation to present today. So as has been mentioned, I'm a postdoc with Monko Lake here in the department. And my postdoc is largely focused on building tools to help with the interpretation of variants in the mitochondrial genome. So this has been a longer standing interest of mine. As was mentioned, I did my PhD on genetic basis of primary mitochondrial disorders. And I've also previously worked as an analyst for clinical mitochondrial genome testing. So today I'm going to talk about navigating analysis of the mitochondrial genome and the process for variant interpretation. And I'll also share with you some of the resources that I've been working on during my postdoc to help aid this process. So I just want to reiterate some of the learning objectives of my talk this morning. So we'll review differences between the nuclear and mitochondrial genomes to help gain appreciation for its unique features that complicate the process of variant interpretation in the mitochondria. And then I'll share with you some of the specific resources that I've been working on, including the gnomAD mitochondrial population database, a tool called MitoVisualize, and a constraint model for the mitochondrial genome that is in preparation. So to start with a little refresher on this part of our DNA. So the mitochondrial genome is a small circular molecule that resides, of course, in our mitochondria, and it encodes for 37 genes, which includes proteins, as well as transfer RNA and ribosomal RNAs. And these all have an essential role in mitochondrial energy generation. So the mitochondrial genome only encodes a fraction of our total genes, but I do want to reiterate the point that even though it's small, the mitochondrial genome has an integral role in human health and survival. And it's widely believed that loss of function of any one of those genes would not be compatible with life in the mitochondria. The mitochondrial genome is, of course, maternally inherited, unlike the biparental inheritance of the nuclear genome. And it also has a number of structural differences. This includes being of circular shape, which reflects its prokaryotic ancestry, as well as some other differences. So, for example, the mitochondrial genome does not include introns, and so splicing is something that is not relevant in the mitochondria. A key difference between the two genomes is their copy number. So in the nuclear genome, there are 2 copies of every gene present in a cell. So one you inherit from your mother and one from your father. But for the mitochondrial genome, there's actually hundreds to thousands of copies of these circular molecules in a cell. And this leads to the phenomenon of heteroplasmy, by where you can have a variant that's present only in a fraction of the total mitochondrial genome molecules that's in a cell or in an individual. And this also means that the mutant load, or the heteroplasmy level of mitochondrial variant, can range anywhere from 0 to 100%. So when a variant is present in every single mitochondrial copy, we refer to this as homoplasmy. And when it's present only in a fraction, we refer to those variants as being heteroplasmic. So we're interested in the mitochondrial genome because variants within it can underlie disease. And specifically, single highly penetrant variants underlie mitochondrial diseases, which are a group of rare, clinically heterogeneous disorders that are notorious for presenting with any symptom in any organ at any age. And this schematic here is showing some of the more frequently encountered symptoms that can be observed in cases with primary mitochondrial disorders. So not only are these presentations clinically heterogeneous, but they're also genetically heterogeneous. So as well as being caused by pathogenic variants in the mitochondrial genome, I also want to acknowledge that they're also caused by pathogenic variants in the nuclear genome as well. So there are some defined clinical syndromes that mitochondrial diseases can present as. That includes Lee syndrome, which is the most common pediatric form of mitochondrial disease. And this is a rare neurodegenerative presentation, so it's often multisystemic. There are also single system presentations, such as the optic neuropathy LON. But while these can present as defined clinical syndromes, many patients don't present with one of these. So really going back to the potential challenge with recognizing a mitochondrial disorder clinically. There are a number of pathogenic variants that have been well characterized in the mitochondrial genome to date. The majority of these are single nucleotide variants, so mostly base substitutions in the transfer RNA genes or missense variants in the protein genes. There are also a large number of large structural variants that have been well characterized, so particularly these large deletions, many of which occur within this region, which is often referred to as the common deletion. So MitoMap is a fantastic resource for mitochondrial genome analysis. And as part of this database, they have curated a list of around 100 variants, which they regard as having a confirmed association with disease. But I think it's widely understood in the field that there are many more pathogenic variants that are yet to be discovered. And something I want to acknowledge is that there is variable expressivity with mitochondrial pathogenic variants. So the same variant can actually underlie multiple distinct clinical presentations. So in illustration of that, for example, is the 3243A to G variant. I'm not sure if you can see my cursor, but this is a variant that is perhaps best appreciated for underlying MELAS, which is a mitochondrial encephalomyopathy associated with lactic acidosis and stroke-like episodes. But it's also been reported to cause maternally inherited diabetes, as well as eye-related presentations and even Lee syndrome. So this can present a challenge when it comes to variant interpretation. So I've started to talk about some of the features which differentiate the mitochondria from the nuclear genome, but there are many of these which further have relevance for variant interpretation, and I'm going to talk a little bit about several of these in more detail now. So I've introduced the concept of heteroplasmy, by where a variant can exist only in a fraction of all mitochondrial molecules. But another aspect of heteroplasmy is that the mutant load or the heteroplasmy level of a variant can actually differ across different tissues in an individual. And this can occur due to random segregation during the developmental process, but also because of selection. So to elaborate on this point, variants in the mitochondrial genome can actually be selected against in dividing cells. So think blood, which is certainly a relevant tissue when it comes to genetic testing. Because if the pathogenic variants are causing a disadvantage, so if they're causing a functional defect, then those variants are selected against. And this causes the heteroplasmy level of those variants to decrease over time. So not only can heteroplasmy level vary over tissues, but it can also vary over the lifetime of an individual. And the best known example of this is that 3243A to G variant. And so what you're looking at here is data, and on the x-axis is the age of the individual carrying the variant, and the y-axis is the heteroplasmy level or mutant load. And hopefully what you can appreciate here is that clearly there is a decrease over time. And it's been estimated actually that this specific variant reduces on average by around 2% heteroplasmy level every year. But what this means is that the heteroplasmy level of a pathogenic variant in the mitochondria can be lower in a dividing cell type like blood compared to a post-mitotic tissue site like muscle in adults. And so what these considerations mean is that the tissue source and the age of the individual from the sample that's being evaluated are important considerations as part of the variant interpretation process. Heteroplasmy also contributes to this phenomenon called disease threshold. And essentially what this is it's a specific heteroplasmy level that a variant needs to reach for disease to occur. And so what this means is that the heteroplasmy level of a pathogenic variant is actually what determines disease status in an individual. And so it's possible that a healthy individual can carry a pathogenic variant and not be affected if the variant is below this threshold. Now, a key challenge is we don't know what this threshold is for most variants. For some of the best characterized pathogenic variants, we do have an approximation for what the threshold is. Typically, we see that a variant has to reach beyond 40 to 50% heteroplasmy level to produce a severe phenotype. But just to give you a feel for the diversity in these thresholds, so at the lower end is something like the 13513 variant in ND5, which can cause Lee syndrome, which is quite a severe presentation at around 40 to 50% heteroplasmy. Whereas at the other end, something like the 8993T to C in ATP6, you typically need to have heteroplasmy levels of above 90% for disease to occur. And so on this point, most pathogenic variants in the mitochondrial genome actually cause disease when they're at heteroplasmy. And some of these are actually never observed at homoplasmy, which likely reflects the fact that that may not be compatible with life. There are some pathogenic variants, though, which only cause disease at homoplasmy and furthermore show incomplete penetrance at homoplasmy. So that includes a number of the variants that have been described to cause the optic neuropathy LON, for example. So haplogroups are another consideration for mitochondrial analysis. And these are groups of mitochondrial variants that are co-inherited, so they're passed down the maternal lineage over generations at homoplasmy. And there are approximately 30 different top-level haplogroups that have been characterized, and everyone's mitochondrial genome can be assigned to one of these top-level haplogroups. And so these haplogroups follow a letter-based system, and they've traditionally been used for ancestry tracking. So if you've done something like 23andMe, perhaps you already know what your haplogroup is. But they also have relevance for variant interpretation, as I'll touch upon. They can represent evidence of being a benign variant in cases. And there's also some evidence out there to suggest that the haplogroup can potentially modify the penetrance of some pathogenic variants, although that's an area that's requiring further research. But speaking of maternal inheritance, so mitochondrial DNA is, of course, maternally inherited. And this means that collecting maternal family history can often be useful if there's to support the basis of mitochondrial disease. However, I did want to touch upon some of the controversial papers that came out in recent years, suggesting potentially that biparental inheritance of the mitochondrial genome was more common than previously appreciated. This has actually more recently been debunked, for lack of a better word. And in fact, follow-up studies suggested that this was likely an artifact of something called meganeumites. So meganeumites or neumites are these regions of DNA that originated from the mitochondria, but have found their way into the nuclear genome during human evolution. And this can mean if you're trying to amplify the mitochondrial genome, you can potentially amplify parts of the nuclear genome by mistake. And that's what led to this report. So to elaborate a little bit more on neumites, they're a really important consideration for analysis of the mitochondrial genome, particularly when you're analyzing types of data where you've got, for example, sequencing reads from both the nuclear genome and the mitochondrial genome. And that's because for new mites, this can cause sequencing reads that actually originate from the nuclear genome to falsely align to the mitochondrial genome. And this can cause false heteroplasmies to occur. And so they're a really important consideration for variant calling in the mitochondria. So on that point, I now want to step through some of the main sequencing testing methods that are typically used for mitochondrial genome analysis. I know that Yale is offering mitochondrial sequencing, and perhaps he can comment on more on that at the end. But there are these three main methods. So the first being targeted sequencing of the mitochondrial genome. And so this is typically employed after something like long-range PCR to selectively amplify the mitochondria. And that can be either via a single large amplicon or multiple amplicons. You can also potentially analyze the mitochondrial genome from exome sequencing data. Most exome capture kits don't provide on-target coverage of the mitochondria. It's typically off-target coverage. However, there are some kits available which do selectively enrich and aim to capture the mitochondria. So it's important to consider which of the platforms that you're working with there. And genome sequencing, of course, is great from the perspective that it provides coverage of both genomes. So there are various considerations to take into account with these different types of sequencing data. One is coverage. So targeted mitochondrial sequencing, for example, has the potential to provide very high coverage of the genome, which is great if you want to confidently call those low fraction heteroplasmic variants where you really want high coverage to be robust. Just to give you a feel for contrast, so potentially you can achieve more than 10,000-fold coverage via targeted mitochondrial sequencing in context with something like a standard exome platform that only provides off-target coverage of the mitochondria. You're looking at more around 50-fold coverage. And in the mitochondrial genome, for something like blood, we often see around 2,000 to 3,000-fold coverage, for example, where you have 20 to 30x coverage of the nuclear genome. So coverage has important implications, as I've mentioned, for the minimum heteroplasmy level that you could call. So something like a targeted mitochondrial sequencing approach, you can potentially confidently call quite low heteroplasmic variants, so somewhere between 1% to 2% and above. Whereas with exome and genome sequencing, the threshold is often a little higher, but not only higher with respect to coverage, but also the consideration of new mites. So if you recall, new mites can lead to false heteroplasmic calls, particularly at those low fractions when you're working with data that contains both nuclear and mitochondrial sequencing reads. So that also is a consideration. So that's, I think, one benefit of going with a targeted mitochondrial sequencing approach is it helps minimize the new mite challenge. So there are some other considerations with this. I won't go too much into depth for time, but I do want to acknowledge that often targeted sequencing of the mitochondria is a separate test. So from that perspective, it often requires clinical suspicion of a mitochondrial disorder to pursue. And mitochondrial disorders, because they're so clinically heterogeneous, can sometimes be hard to recognize. And indeed, there have been a number of studies coming out in the last few years where groups are going back to their large unsolved rare disease cases and finding mitochondrial variants that were missed because these were cases that weren't suspected to have primary mitochondrial disorders. So showing the potential utility of follow-up of the mitochondrial genome in broad unsolved cases. So there are actually a couple of years ago now, some specifications were published for adapting the ACMG guidelines for variant classification to the mitochondrial genome. Because as I've alluded to already, the mitochondrial genome has a number of unique features which are important to take into account when you're assessing variants within it, and which means that you need a specialized process to do this. So just to refresh, the ACMG guidelines are those where they have a set of criteria and you utilize them to determine a classification for a variant that you found in a patient, ranging from pathogenic, so that is, it is the cause of the patient's disease, to benign, or when there's not enough evidence to confidently assign either of those classifications, they're of uncertain significance. I'm not going to go into this in too much detail, but just to elaborate that these criteria provide differing levels of evidence for either pathogenic or benign status, ranging from support to a strong level criteria across different lines of evidence, ranging from population and computational data to segregation data, among others. I don't have time to go into the criteria in too much detail, but I did want to point out some of the examples of how these criteria have been modified for its application to the mitochondria. So first of all, just going over some of the benign criteria. So BA1 is actually a standalone benign criteria. So if a variant satisfies this, it can be regarded as being benign. So in the nuclear genome, it's recommended that any variant that has a population frequency of more than 5% can be regarded as benign. But in the mitochondria, they've suggested a different approach. So for example, they've suggested that a top level haplogroup variant, if you find it in an individual of the same haplogroup, that can be regarded as being benign, or variants that have a population frequency of greater than 1%. They've also provided some other guidelines for allele frequency. So for example, anything above 0.5% heteroplasmy level can be assigned strong evidence of benign. And they've also modified how segregation data can be used to provide benign support, and also taking account heteroplasmy levels as part of that segregation. So for example, observing a variant at a higher heteroplasmy in a healthy adult. So it's higher heteroplasmy level in that individual than in your patient, particularly if it's in the same tissue as what you've tested in the patient, can potentially be used as strong evidence of benign support. So just to touch upon some of the pathogenic modifications. So they've also provided some guidelines on how you could use segregation data to provide evidence of pathogenicity. So for example, a variant that's co-segregating with disease in at least 2 to 4 family members, maternal family members, as well as the heteroplasmy level is segregating, can be used to support. And they provide some guidelines about how to factor in heteroplasmy levels and tissues tested as part of this. There are also some criteria that they've recommended to be removed. So one of those includes, for example, applying criteria for variants that are in well-established functional domains. This is partly because there aren't well-established functional domains in the mitochondrial genes, particularly the mitochondrial proteins. And so for that and other reasons, they've recommended not using that criteria. They've also suggested a slightly different allele frequency for providing evidence of pathogenicity. So in the nuclear genome, it's suggested that it should be absent from the population databases to provide evidence. But in the mitochondrial genome, they're suggesting that an ultra rare frequency of around 1 in 50,000, that any variant below this can be assigned pathogenic supporting criteria. And lastly, they've also expanded upon some criteria to help enable their application more easily to RNA variants, given that the ACMG guidelines are predominantly for missense for protein variants, sorry. So for example, what they've put forward here is that in an RNA gene, if you see a variant and it occurs at a position where a different pathogenic variant has been reported, then that can potentially provide support of pathogenicity. So they've also provided a specification of what in silico predictive tools can be used or should be used in their opinion for classification. And I just wanted to comment on this because they've only suggested use of one specific tool to predict the effect of mitochondrial missense variants and 2 tools for transfer RNAs and none for ribosomal RNAs because there actually aren't any available. So they've recommended this because most of the tools that you're probably familiar with, like Polyphen and SIFT and CAD, they're actually built and trained upon variants from the nuclear genome specifically. So they don't actually apply very well to the mitochondrial genome. So on that point, there's actually a relative paucity of tools for interpreting variants in the mitochondria because most of the tools that as human geneticists we're used to working with are actually really not suitable for this genome. And not only with predictive algorithms, but also population databases. So most of the population databases out there only include nuclear variation and are missing mitochondrial variation. There are some mitochondrial population databases out there. And so actually at the time that those ACMG guidelines were put out, it was the MitoMap population database that was the predominant one used for variant classification. And this is a fantastic resource, but it does have some important caveats that are worth noting. So for example, it uses publicly deposited data that's of varying qualities and varying sources. So for example, it includes samples that are known to have disease and or be from cell lines or cancer samples, for example. So I think it's the experience of many that we're finding that many variants that we find in patients are ultimately falling out as being variants of uncertain significance. So there's just not enough evidence available out there to confidently assign pathogenic or benign status. And so really, I think many others agree that really we need some tool development to help catch the mitochondria up to where the nuclear genome is and to help address resolving this burden of uncertain significance. And so on that point, I'm now going to transition to telling you a little bit about some of the resources that I've been working on with others as part of my postdoc to help address this gap. And so I'm going to start by talking about the gnomAD mitochondrial population database. So gnomAD is a database of genetic variation that's observed in the human population, and it's been widely used for variant interpretation in rare disease cases. And so historically, it only included nuclear variation, but actually it was the availability of whole genome sequencing data that really opened the door for being able to expand this to also include mitochondrial variation. So together with our collaborators at the Broad Institute, we called variants in the mitochondria in more than 50,000 individuals for whom genome sequencing data was available. Most of these samples, I will say, are from blood. We don't have the tissue source for all of them, but for those that we do have, most are from blood. But this resource actually represents one of the largest mitochondrial population databases available and the first large-scale database built from whole genome sequencing data. Another key utility of gnomAD is that it's depleted of cases known to have severe pediatric disease, which means it's a really useful reference set for allele frequencies. I'm not going to go into detail, but I do want to acknowledge that a new pipeline was built as part of this project and used to call the variants in the mitochondria from this whole genome sequencing data. That is also freely available as part of the GATK tool suite. So we really undertook quite stringent quality control as part of the development of this database, given that it was going to be, or we knew it would be used as a reference set of population allele frequencies. And this included sample level filters as well as variant level filters. And something we were really cognizant of trying to avoid was those false heteroplasmy calls that can occur due to new mites. So again, to refresh, that's when sequencing reads that are actually from the nucleus falsely map to the mitochondria. And so from our analysis, we found that samples that had low mitochondrial copy number. So that's the relative ratio of mitochondrial molecules to the nuclear genome, as well as variants that had low heteroplasmy levels, that these were highly enriched in these false positive heteroplasmy calls. And so we adapted a number of filters to minimize that. And as part of that, we were left with around 56,000 samples. And we also made a decision to only release variants that had a heteroplasmy level of above 10% as well. So this was quite a conservative approach, but we really wanted to avoid any heteroplasmy pseudonimites. So this call set includes data on homoplasmic variants, as well as heteroplasmic variants. And this is fairly significant because population databases like MitoMap prior to the advent of gnomAD hadn't included heteroplasmic variants, whereas genome sequencing data gives you the ability to provide that. So taking a look, I think to just emphasize the power of a data set like this, we observed variation at more than 50% of all bases in the mitochondrial genome. So even at these sample sizes, we're really starting to really dive into the possible population variation that exists in this genome. Most of these variants were single nucleotide variants and observed at homoplasmy, but we did see around 15%, for example, that we only ever saw at heteroplasmy. So only present in a fraction of all molecules. And so these are a group of variants that would have been missed by previous approaches for mitochondrial databases. The majority of the variants were rare in the population. So around a third we only observed in one individual. And more than 2/3 were present at less than 0.1% allele frequency. Of those observed above that level, they were nearly all haplogroup defining variants. And we can see that the singletons are highly enriched in heteroplasmies as well as non-haplogroup homoplasmies as well. So diverse ancestry is a really important consideration of a population data set. And so we were definitely interested to assess the spread of diversity, mitochondrial ancestry in gnomAD. So to refresh, everyone's mitochondrial genome can be assigned to one of around 30 top-level haplogroups, which originated with the mitochondrial Eve in Africa and evolved over evolution. But they can broadly be categorized into being of African, Asian, or European ancestry. And so when we looked across gnomAD, we saw that we had quite good coverage across mitochondrial haplogroups and ancestries. We were missing a couple, but we had nearly all represented. And another observation I just wanted to briefly comment on was that we noticed that the number of homoplasmic variants correlated with the haplogroup of the sample. So just to orientate you, the reference genome for the mitochondria belongs to haplogroup H. And so what we could see is that as the distance from the reference haplogroup in the phylogenetic tree increases, so does the number of variants. So individuals of African mitochondrial ancestry have anywhere between 50 to 100 variants, those of Asian mitochondrial ancestry between 25 to 50, and European around 0 to 50. We then took a closer look at known pathogenic variants in gnomAD. And to do that, we focused on the 100 or so variants that have been curated by MitoMap as having a confirmed disease status. So we saw most of them weren't present in the gnomAD dataset, and we saw only around 30%. So what you're looking at here is the dot plot, which shows the pathogenic variants that we did see in gnomAD and their heteroplasmy level, which is across the x-axis. So what we can see here is that most of the individuals that have a pathogenic variant are carrying it at low heteroplasmy levels. But we do see some instances where we are seeing some of these pathogenic variants at homoplasmy. But when we take a closer look, most of these variants we're seeing at homoplasmy are known to be associated with incomplete penetrance at homoplasmy and or associated with adult onset conditions, so like deafness or optic neuropathy, which I think explains why we're seeing them in gnomAD. And when we amass this data, we calculate that there is a total carrier frequency in the population of around 1 in 250 individuals for pathogenic mitochondrial variants. So just to remind, because we were only releasing variants above 10%, this is a carrier frequency for variants above that threshold. And furthermore, represents a lower bound, given that there are many more pathogenic variants to discover, and also that there will be individuals carrying these variants below 10% as well. So I think it just really emphasizes the total carrier burden in the population for pathogenic variation. We also took a closer look at the allele frequencies for these variants to see how they aligned with the ACMG criteria. And so what you're looking at here are the allele frequencies for the pathogenic variants we saw, categorized into different groups, and they're colored by their disease association status. So those that are only reported to cause disease at homoplasmy and are never, at heteroplasmy, and are never seen at homoplasmy are colored in orange, and those that are known to cause disease at homoplasmy are in blue. But what we can see here is for the pathogenic criteria, 90% of variants have a homoplasmic allele frequency that is meeting this pathogenic criteria. So I'm looking at the homoplasmic allele frequency because the guidelines from ACMG are derived from MitoMap, which is homoplasmic variation. And we can see that actually all of the variants that are only associated with disease at heteroplasmy meet this threshold. And it's only really those that are associated with incomplete penetrance that don't. We also see, importantly, that none of these variants have an allele frequency that would satisfy a benign criteria. So they're all below 0.5%, including when we just look at haplogroup frequencies or population-specific frequencies for those calculated from nuclear variation. We did also look at heteroplasmic allele frequency. It does look somewhat similar to homoplasmies, that the majority of variants do also technically fall below this, this threshold recommended for pathogenic criteria. But I will say that the clinical utility of heteroplasmic allele frequencies is unclear. gnomAD and other data sets with heteroplasmy information wasn't available when the ACMG criteria were developed. So this is something where an update of those guidelines will be needed to guide if and how that data should be used. So the data is freely available via the browser. So you can see it provides homoplasmic and heteroplasmic allele frequencies. I also wanted to note that you can also browse variants that we filtered out due to various quality control checks. And something that we commented on in the manuscript was that we do recommend caution against using the absence of the variant in gnomAD as evidence of pathogenic support. Because it's possible that we actually did call it, but it was filtered out due to one of the various filters we applied. And so interrogating the filtered variants can be useful for that. There's also more information that can be available for specific variants, including population and haplogroup frequencies as well. Okay, I'm now going to talk a little bit about MitoVisualize. And this is a tool that was really motivated by my own experience analyzing clinical mitochondrial genome tests. And this was specifically built to facilitate the analysis of variants in the RNA genes, as well as the circular structure. So I'm just going to step through some of the key functionalities of this tool, but it's freely available right now. You can Google MitoVisualize or go to mitovisualize.org and have a play with the tool yourself. So the first key functionality is to query a variant of interest. So for example, a variant you found in a patient that's within either a transfer RNA or ribosomal RNA gene. And this is a screenshot of a query for the 3243 A to G variant. And so what you can see here is it's showing the location and the effect of the variant in the RNA secondary structure. And this is actually a useful piece of information for variant classification, because there are some specific areas of the transfer RNAs that are known to particularly be enriched in deleterious variation. We also provide a suite of various annotations that are useful for variant interpretation, including population frequency, disease association status, and their in silico predictions, with hyperlinks to the data source so you can interrogate further. But you can also freely download this schematic, which shows the position of the variant in the secondary structure for reuse in publications or presentations as well. We also provide functionality to visualize data across the secondary structures. So here's a screenshot showing that for variants associated with the disease in one of the transfer RNA genes. So if you recall, this can be a useful piece of evidence for variant classification, for example. And again, you can download these figures. And the third key functionality I wanted to cover was the ability to label or visualize a base or a region in the mitochondrial genome. So here's an example of that, visualizing a large deletion that was identified in a patient. So we can visualize the location of that in the structure, as well as the genes encoded by the region, and links out to the MitoBreak database, for example. And again, these figures can be downloaded and incorporated into presentations and publications describing variants of interest. So if you're interested in more information, that's available on the About page on the site, and you can also browse the article which describes this. But if you have any feedback, if there's any features that you'd like to see on the tool that isn't there, we would love to hear from you, so feel free to reach out. Okay, so the last tool I'm going to present to finish off my presentation today is a mitochondrial constraint model that we've been working on. So what is constraint? So constraint models are techniques for identifying exactly where in the genome negative selection is removing variants from the population. And so constraint models have been developed for the nuclear genome and have been widely useful, but not yet available for the mitochondria. And so when I'm talking about constraints, I'm talking about genes and regions when the observed level of variation in the population is less than expected based on a mutational model. And the reason that these have been highly useful for variant interpretation is because these constrained regions are enriched in pathogenic variation. So population data sets like gnomAD have been used to develop constraint metrics for the nuclear genome, and these have been widely used. And so the development of the mitochondrial population database really provided a great opportunity to go ahead and develop a framework for assessing mitochondrial constraint, because the nuclear framework couldn't be applied because of all of its unique features. So we went ahead and did that. To put a lot of work into one neat sentence, we built a mutational model that we could use to calculate the expected level of variation in a mitochondrial gene or region in gnomAD. And when I'm talking about calculating expectation, I'm talking about under neutrality, so under the assumption that there's no selection occurring. So we could do that and then compare it to the observed level. So to just help orientate you with this concept, what you're looking at here are the results of observed compared to expected variation in each of the protein coding genes in gnomAD, focusing on synonymous variation. So what this is showing is that the observed levels are approximately equal to expectation, which is consistent with the fact that synonymous variation is not expected to be subject to strong selection. In contrast, we can see missense variation is falling off the line. And in fact, what we're seeing is that there is far less observed missense variation than that expected in most genes. And this is consistent with selection removing many missense from the population. And in contrast for stop gain, essentially what we're seeing is a near incompatibility of loss of function variants with life in the mitochondrial genome because we observe them at such low levels compared to expectation. So we can use this framework to explore and quantify the relative strength of selection against different groups of mitochondrial variation. And so that's what you're seeing here for various functional classes of variants across the mitochondria. And here we're expressing it as a ratio of observed to expected. So the closer to zero, the more constrained the group is. And what we can see is that the ratio generally correlates with the predicted severity of the group. And there's a couple of observations I want to point out from this. The first is that RNA variation appears to be subject to a similar level of selection as missense. And this is particularly interesting for the ribosomal RNA variants, which are typically overlooked in disease analyses, partly because there isn't any in silico tools for predicting whether they're likely to be deleterious or not. But this suggests, as well as some other analyses, that we're likely missing pathogenic variants in these genes. We also see partial depletion of intergenic or non-coding variation. And again, in the interest of time, I won't go into this, but I will say that we believe that this is driven by selection against variants in annotated non-coding elements, which have a role in transcription or replication of the mitochondrial genome. And so we think that there may also be some pathogenic variation that's hidden within the non-coding functional elements. We also took this approach looking at disease-associated variation in ClinVar and MitoMap. And again, what we can see is that the ratio approximately correlates with classification. So ranging from benign, which has a ratio of approximately 1, to pathogenic, which is close to 0. So this shows that our approach is also able to separate between pathogenic and benign variation. And just to briefly add that the error bars that you're seeing are 90% confidence intervals around the ratios. So the last piece that I wanted to share with you is one of the specific analyses we've done as part of constraint, and this has been looking at regional missense constraint. So regional missense constraint is a phenomenon by which specific regions in a gene can be more constrained than the gene itself. And this is a concept that has been well studied in nuclear genes, and more specifically, it's also been shown in the nuclear genome that these areas of regional constraint are enriched in pathogenic variation. So they're useful for variant classification. So therefore, we were highly motivated to go ahead and also look at this in the mitochondrial genome. And so we developed a method for this, and we identified that approximately 15% of the protein sequence was regionally constrained, and specifically regionally missense constrained. And when we mapped this back to the 3D structures, what we observed was that even though they were spread out in the linear sequence, they appeared to be clustered topologically in the 3D structure. And in fact, when we dug through the literature, we found that for this specific gene example, ND1, this is clustering within a binding pocket for quinine, which is a cofactor for the enzyme that this protein belongs to. So it would have an essential role in function. And so we think this data can also be helpful for illuminating which areas are most important for function, which we think will have utility given the lack of annotated functional domains. So we have performed odds ratio enrichment analysis using missense variants from ClinVar and MitoMap, and shown that we can see that their pathogenic variants are enriched in these areas of regional constraint, and that they're enriched to a lower extent in residues that are very close to regional constraint in the 3D structure. And when we assessed across a group of variants that were curated by a single clinical genetic service, in cases that were suspected of primary mitochondrial disease, so this is actually the service I used to work for, what we could see is that the proportion within or proximal to regional constraint also correlated with classification. So for example, we can see pathogenic and likely pathogenic have the highest proportion within regional constraint compared to benign and likely benign, where there is actually not any in regional constraint. So this corroborates the data from ClinVar and MitoMap. So I'm going to finish there, and just to reiterate some of the key take-home points. So the mitochondrial genome has many unique features which are important to consider for variant interpretation and analysis, because it dictates that a specialized process to be undertaken. And that there are new tools and resources that are currently being developed, and hopefully this will improve variant classification in this genome. There are many people that have contributed to the work that I've presented on today. I just want to take a moment to acknowledge them all, but especially my mentor, Nicole, who has really been instrumental in this as well. Very happy to take questions and discussion. Thank you.
Thank you, Nicole. You have a wonderful talk. Yeah, that open up for the question or comments from anyone. You can unmute yourself if you have a question.
Hello, my name is Ivan. I'm a postdoc from Stephanie Nicole's lab. Hi, Nicole. It's very nice talk. I really like it. Really enjoy it. I have a question about this mitochondrial variants. Do you know these variants can be triggered by any genome genes activity, and then like leading them have these mitochondrial variants?
So just to check that I confirm, are you saying can variants in the nuclear genome cause variants to be created in the mitochondrial genome?
Yeah, basically I'm thinking about like some co-activator from the gene expression, and then they can, because you say there's the disease threshold in this mitochondrial variant, and it happen in certain time in certain organs. So I'm thinking if there any things like trigger this activity, like a threshold, and then suddenly make
Oh, I think I see what you're saying. So is there anything that can cause mitochondrial dysfunction to begin to occur? Any triggers for that? Yeah, so there likely is. That could be environmental factors. Also things like illness are known to exacerbate mitochondrial presentations, and also energy demands over development. So research has shown, for example, that with Lee syndrome, which is a pediatric presentation, that symptoms typically onset or progress during periods where brain development needs a lot of energy. So there are certainly other factors that can contribute there. And it's likely to be tissue or organ specific, as you acknowledged.
And another question, sorry. Also, when these mitochondrial variants happen, is there any mitochondrial phenotype we can observe in this patient or in this mice model and any model mitochondrial phenotype?
Are there animal models for mitochondrial variants?
No, I just wondering if these variants happen, is there any mitochondrial phenotype or function changed? Do we know previously?
So there are so mitochondrial variants can cause functional defects and phenotypes, including in animal models, if that's answering your question. There are some animal models. You can also create cellular models now. Those tools have only very recently become available. So there are now base editors, for example, where you can create specific certain types of mutations that you can characterize in cell lines. So they have limitations. But there is the ability to study the functional effects of mitochondrial variants.
Thank you very much.
Thanks.
Hi, Nicole. Can you hear me? Yeah.
Yes.
Okay. Yeah, this is Penny Lee. I kind of noticed you mentioned the paternal mitochondrial issues. And I think it's from some basic research, is like in animal models, if there's a paternal mitochondria into the eggs, and then it will trigger apoptosis, and then that get rid of that as a protection. Any progress in the human aspect for that part?
For the assessing the contribution of paternal mitochondrial inheritance? Yes, so there has been some papers in recent years, which I alluded to. So there was a paper several years ago published in PNAS, which suggested that biparental inheritance of the mitochondrial genome may be more common than previously appreciated. However, more recent studies have come out since to suggest that those results were likely a technical artifact of new mites. So they were caused by regions that were nearly identical or highly homologous between the two genomes. So I think the consensus in the field is that it's still a nearly exclusively maternal inheritance, although there are some very rare examples of paternal inheritance that have been reported.
Okay, thanks. Yeah.
Hey, Nicole, this is Chen from the DNA lab. I have a question. Hi. So yeah, so mentioned about this, I think the carrier frequency for a pathogenic variant in the mitogenome is about 1 out of 250. So what is the comparable rate for the nuclear genome? I thought people always talk about this, the mutation rate is higher in mitogenome as compared to nuclear genome. Do you have any comments about that?
Yeah, that's a great question. I know there was a study published a couple of years ago which assessed the carrier frequency in a subset of nuclear encoded mitochondrial genes, but I can't recall that number off the top of my head. I know as a group, it's estimated that mitochondrial disorders have a prevalence of around 1 in 4,000 to 5,000, though it's likely more common than that. But as you said, the mutation rate in the mitochondrial genome is actually an order of magnitude higher than the nuclear genome, so at least 10-fold higher. So if I had to guess, I would say the carrier rate is much higher in the mitochondrial genome, though I can't recall the exact numbers off the top of my head.
Yeah.
Great. Any other comments or questions? Kind of last question. Nicole, for the gnomAD mito population, do you happen to look at whether it's age specific or the mutation rate, like a aging process actually increase the allele frequency?
Yeah, so also a good question. So we don't have in gnomAD, age isn't available for every sample, but there is an increase in the number of heteroplasmies, for example, as people age. That's something that's been well studied and reported. And so that reflects actually, I didn't get into this detail, but heteroplasmies, for example, can be a mix of those that are inherited, but also some that occur somatically or de novo. And so we are also likely capturing some of those in the data set, and that's why there's likely to be an increase over age with that. There is, if you are interested in a specific variant, you can actually assess the age distribution of carriers as part of the gnomAD database. So for where we do have the data, we do make it is made available via the browser.